Welcome and thank you for joining us today for another Virtual Investor KOL Connect segment featuring Sonnet BioTherapeutics. My name is Janine Thomas. I am CEO of JTCIR and I will be today's moderator. I am very pleased to be joined by members of the Sonnet BioTherapeutics team. We have Dr. Pankaj Mohan. He is founder and chief executive officer, and Dr. John Cheney. He is co-founder and chief scientific officer. We also have two prominent key opinion leaders. We have Dr. Andrew Handafar. He is with Cedars-Sinai Medical Center, and Dr. Sant Chawla, Director of Sarcoma Oncology and Research Center. So today we are here to discuss pancreatic cancer and Sonnet's non-dilutive investigator-initiated collaboration announcement related to SON 1210. Before we get started, I want to remind our audience that Sonnet BioTherapeutics is publicly listed on NASDAQ and trades under the ticker SONN. And during today's discussion, the company will be making forward-looking statements, and I encourage everyone to view the company's website at sonnetbio.com and the SEC's website for the latest information. Dr. Mohan, to set the stage for today's discussion, can you describe the collaboration agreement you announced today?
Sure, Janine, it's a pleasure talking about this here. Sonnet entered into a collaboration agreement to commence investigator-initiated phase 1/2A study of SON 1210 for the treatment of pancreatic cancer. I'm very excited by this collaboration. Sonnet was approached by key opinion leaders, Dr. Chawla and Dr. Handafar. They proposed using our first bifunctional molecule, SON 1210, a unique synergistic combination of interleukin 12 and interleukin 15 on our platform, which is targeted to cancer tumor for an investigator-initiated clinical study. This platform is especially relevant to cancer tumors that overexpresses a protein called SPARC, which is a homing mechanism for a platform. Pancreatic cancer is one such cancer tumor that overexpresses this protein called SPARC.
Dr. Handafar, we are grateful to have you with us today. As additional background, can you please discuss pancreatic cancer, the patient journey, including the standard of care and limitations with current treatments?
So pancreas cancer is becoming a more and more serious problem. The incidence, unfortunately, is growing in the United States and worldwide. In addition to that, the treatments have been traditionally not very effective. We have difficulties diagnosing it early. We have difficulties with some of the imaging modalities, but most importantly, our treatments are incredibly insufficient. The standard of care of pancreas cancer really hasn't changed for 25 years. It's chemotherapy, and the chemotherapy has not been as effective as the patients or the physicians would want. Really, the next step for us is to be able to harness the ability for the immune system to fight the tumor and to develop better immune therapies for this difficult disease. In regards to immune therapy, we believe that SON 1210, a novel bifunctional IL-12, IL-15 fusion protein, is a promising new immune therapy for pancreatic cancer that has the opportunity to improve outcomes.
Dr. Cheney, it would be great for you to discuss the platform technology and how SON 1210 works.
Clinical trials using cytokines or interleukins have had limited success in clinical trials. And this is based on the small molecular weight of these interleukins, resulting in a very short half-life in the body of the patient, and also are not tumor-targeted delivered. Sonnet has developed a fully human albumin binding platform that specifically addressed these questions. We discovered this single-chain Fab construct through using a phage library of human single chains. It was designed to specific physical properties of the single chain based on tumor biology, and that is that albumin accumulates in solid tumors. In the figure shown on the screen, on the upper left-hand corner is a cartoon representation of the Fab construct binding to albumin. Red and green is the Fab single chain. on each side is linked. IL-12 on the left side and the right-hand side is IL-15. When this complex is formed, it shows no steric hindrance of a biological activity of IL-12 or IL-15. The Fab binds to albumin over a wide pH range of 6 to 7 because tumors are shown to be acidic in nature, and we bind in the lower nanomolar region. Albumin, as you can see in this cartoon structure, has numerous types of binding domains. The three of them that we are interested in is the binding to the FcRn, which increases circulation time within the patient. It also has two other domains binding to GP60, which is an albumin receptor expressed in tumor vascularity, along with SPARC in the TME itself. The cartoon represents the binding of this entire complex. There's no interference of binding to SPARC, GP60, or FcRn when bound to our Fab construct. The MOA of this is shown in the left-hand lower corner, where Sonnet 1210 is injected into the patient, circulates into where it gets into the capillary beds within the tumor itself. The capillary beds overexpress FcRn and GP60, again, which our albumin Sonnet 1210 binds to those receptors and is transported into the tumor microenvironment. We have shown in 3 different in vivo tumor models that we have a 4 to 5-fold increase accumulation in the tumor environment when the cytokines are bound to the Fab construct. So we have tumor penetration. Most importantly is tumor retention. That is based on this complex being able to bind to SPARC. SPARC is overexpressed in numerous tumors, but especially in pancreatic cancer. It's been shown that pancreatic patients have extremely high levels of SPARC present, and that has been correlated to the survivability of the patient. The higher the SPARC in pancreatic patients, the shorter time they have after a diagnosis. So we have essentially shown that it binds to SPARC at 10 nanomolar within the tumor microenvironment. Also, which is important, is the FcRn receptor in the tumor microenvironment. There was a paper published in 2020 which looked at 10 different human tumors. 8 of them showed that FcRn is overexpressed in the tumor microenvironment, capillary beds, and pancreatic cancer showed about a 50% increase in this FcRn expression. So hence the mechanism of action proven is enhanced PK, tumor penetration, and retention of the tumor with the Fab construct. The Fab construct is shown on the right-hand side of this figure, in that the Fab construct can either have 1 or 2 interleukins attached to the albumin binding domain. We specifically chose 12 and 15 as our first interleukin bifunctional construct, and that is based on the synergistic functionality between these 2 interleukins. IL-12 increases the IL-15 receptor, increases FcRn, the polarization of M2 to M1, increases NK cells and T cells, increases Th1, and most importantly, decreases Tregs, which we have shown in FACS data in our animal studies. IL-15 increases the IL-12 beta receptor along with NK cells. But the most important functionality of IL-15 is that it prevents CD8 memory cells from undergoing apoptosis. So hence the survivability of these memory cells increases the immunosurveillance long-term on these patients, preventing the reoccurrence of tumor at a later time. So hence we thought that this would be our best combination for our first bifunctional molecule. So in summary, IL-15 and IL-12 have been used in mouse tumor models and have shown to have efficacy in pancreatic cancer. We have used this construct in combination with chemotherapy. chemotherapy kills cancer cells, but it also has a negative impact on immune cells. So hence, we are doing something different in our phase 1 trials in that we are doing staggered cycling of chemotherapy first, giving the immune cells time to recover before we stimulate with the 12-10 complex, then dosing later back with the chemotherapy, then alternating with the immunostimulator. We feel that the combination of this unique bifunctional molecule of 12-15 in combination with chemotherapy may have a significant impact on cancer patients with pancreatic cancer and other cancers, have an increase in survivability of these patients.
Now on to you, Dr. Chawla. Thank you for joining us. Can you share some background on your motivation to explore this collaboration and why you chose to work with SON 1210 in pancreatic cancer, as well as your development plan for the phase 1-2A program?
Thank you, Janine. As we all know, cancer is a very difficult problem, and I have been always interested in the albumin binding particles which are preferentially taken by cancer. And some of the drug, this technology and platform and this molecule. So this Sonnet molecule is interleukin 12 as well as interleukin 15, which is bound to the albumin. We already have number of drugs approved, including Abraxane, which is albumin-bound Taxol and is superior to regular Taxol. And I was involved in the clinical trial of a drug named rapamycin, where rapamycin was bound to albumin and showed superior results. Similarly, there was another anthracycline, aldoxorubicin, and that was found to be superior because of albumin bound. Cancers are hungry for albumin, and once it leaks through the system, the drugs are taken preferentially by the tumor. So this drug, interleukin 12-15, was very interesting to me. First, the platform. Number two, interleukin 12-15, both are immunotherapy and have been shown in the preclinical and even clinically previously promising results, but had significant amount of toxicity, and this albumin bound has gotten over the major toxicity. I had personal experience in the phase 1 and phase 2 of the interleukin 12 in number of solid tumor patients and carcinoma of ovary. Some of the patients are impressive responses and long-term control of the disease, and pretty much very minimal toxicity confined to the little fever and fatigue. So based on the innovative technology, albumin bound and immunotherapy, it excited me to do a clinical trial in pancreatic cancer. And pancreatic cancer itself is highly lethal, no immunotherapy approved, and its microenvironment is deficient in immune system. And so this technology and high expression of the spark, which is the albumin-mediated mechanism, we think the drugs will be taken over. And that was the motivation. And so in Santa Monica area with my team, and I invited my other colleagues from City of Hope, Dr. Chung, who is a renowned and expert in pancreatic cancer, Dr. Handifar, in similar league, he's from Cedars-Sinai, and Dr. Jeff Weinberg, who wrote recently a new protocol and approval, Napoli, which is the standard of care for pancreatic cancer. I requested and invited them to join in this venture for pancreatic cancer.
So back to you, Pankaj. This is exciting for the pancreatic cancer community and certainly for your pipeline. I have to ask, can you share why this collaboration makes sense right now? And also, you have stated you were in pursuit of a licensing agreement. Make that plural, agreements. Has that strategy changed?
Janine, let me take the second part of the question first. We remain focused on identifying a partner or partners for our late stage assets where we now have early clinical data for both SON 1010 and SON 080. We are working hard on advancing our conversation with SON 1010 with the data that we have, but we realize it takes time. Following recent strong data, we have also commenced BD strategy discussions on SON 080, which is focused on diabetic peripheral neuropathy, a largely high unmet medical need. This present collaboration we are talking about came from left field and opens up new opportunity. As you know, and I want to emphasize that our SON 1010 positive data was key for this unsolicited collaboration. In summary, data is what gets a deal. Further, this collaboration is a non-dilutive mechanism to progress a key oncology asset in a largely unmet medical need of pancreatic cancer with renowned leaders in this space. We believe that the data from this clinical trial has the potential to add to our growing body of data for our platform and enables us to explore SON 1210 as a combination therapy and ultimately be leveraged for licensing for other combinations other than chemotherapy. Following the completion of the upcoming study and with the right data in hand, we believe that we will have the line of sight into the next phase of development, as well as we believe it could provide us with the opportunity to begin having conversations with potential licensing partners to discuss SON 1210 with checkpoint inhibitor, CAR T, CAR NK, etc.
So, Dr. Chawla, before we close, what are the next steps for the development program?
The program initially is this new molecule is a combination of interleukin 12 and interleukin 15 on the albumin bind one molecule. And they both are synergistic and enhance the effect of both. We know the standard of treatment or best treatment for frontline pancreatic cancer is Napoli, which is a combination of liposomal irinotecan chemotherapy. So our intention is provide the best care available now and then add this immunotherapy, which are two immunotherapy, which are albumin bound and albumin preferentially taken in pancreatic cancer through GP60 and spark protein. And then it makes cold immunogenic environment of pancreatic cancer hot. So the chemotherapy will do its part, hopefully kill the tumor, expose the antigen, and these interleukin 12 and 15 come in, boost its further cancer killing. So 1 + 1, we are hoping will make 3 or 4 than just 2. And fortunately, I congratulate the company that this albumin bind technology has significantly decreased the toxicity, at least in the interleukin 12, and we expect the similar in this new molecule interleukin 12-15 combo. And so it will be that we will initially do a phase 1 study, which takes about 20 to 30 patients in dose escalation, find the MTD or maximum tolerated dose or approximately, and see the initial activity and toxicity. And once we achieve that dose, then we will be treating untreated pancreatic cancer along with the frontline protocol as Napoli recently published by Dr. Jeff Weinberg. We are seeking that this is a consortium which is innovative immuno-oncology consortium for pancreatic cancer. Dr. Chung from City of Hope, Dr. Hendifar from Cedars-Sinai, and Dr. Jeff Weinberg from UCLA. We all embark on this voyage in frontline pancreatic cancer, which is a deadly disease and big unmet need in this cancer. We hope that we will be able to start the complete phase 1 by middle of next year, but start in next few months as the company is ready to file the IND, investigational new drug, with FDA. And as soon as we do the phase 1, middle of next year, hopefully the combo trial will be started.
Pankaj, any closing remarks?
As you heard from KOLs, that pancreatic cancer is a high unmet medical need. I am very excited that KOLs have chosen Sonnet 1210 to be a prospective candidate for the next generation pancreatic cancer care. This collaboration, I believe, opens up new avenues for pipeline advancement in a non-dilutive way, which we believe has opened up a window of opportunity for us to leverage this asset 1210 into multiple ways. Thank you so much, Janine.
With that, this concludes the Virtual Investor KOL Connect featuring Sonnet BioTherapeutics. I would like to extend a huge thank you to Drs. Pankaj Mohan, John Cheney, Andrew Handifar, and Sant Chalo for joining us today. I'd also like to thank our audience for your time and attention. As a reminder, Sonnet BioTherapeutics trades on NASDAQ under the ticker SONN. And if you like what you saw today, I encourage you to visit sonnetbio.com for more information on the company and to sign up to follow their alerts, as well as follow their social channels to stay current on the latest information. I'd like to thank everyone again and wishing you a great rest of your day.